Hello everyone. Today we will learn about the experiment on how to prepare herbarium. So the objective of your experiment is uh, the preparation of herbarium, and the outcome is that upon successful completion of this practical, you will be able to describe the method of preparation of herbarium, which is mapping with your course learning outcome too in your syllabus. So let's see what is herbarium. So herbarium is actually a collection of the plants. So it's a kind of a specimen, which can be of plant, which can be of any other living being. But since we are dealing with the herbal plants, so we will discuss about plants. So it is a collection of plant that has been preserved and dried according to the predetermined standards and separated by or sorted by the labels. So herbarium is actually a specimen which may have the twigs or leaves, flower, fruits, or other part of the plant, and in a preserved manner, which is of course is dried and preserved and stored. But all these procedures are done according to the predetermined standards of herbarium preparation. So what are the requirements? So these are the requirements for preparing the herbarium. Like we need large plastic bags, we need notebooks and pencils, we need plant press, or we call herbarium press, recycled newspaper, cardboard, glue, needle, thread, and we need plastic sleeves and magnifying glasses and covered hot air oven, and also some preserving sheets. So this is these are some of the pictures of the requirement what we need. Okay, as you see here, we need cutter, knife, cleaver for cutting the plant parts from the field. So this preparation is done. Herbarium is prepared once we go to the field and collect the sample from the field, and then we bring it to the lab and then prepare the herbarium. So these equipments are required for the collection of the sample. And of course, we need a tag for labeling, because when we are collecting so many samples, when we come back to the lab, we may not remember. So it's always good to put a tag or label for each collection. We need shovel for maybe digging out the some of the plant parts. Let's say it's a rhizome or roots we want to preserve. We need plastic bags and this Caesar notes, pencils. We need it in our collection during the collection of the plant sample in the field. Okay, so what are the steps involved in the preparation of the herbarium? So these are some of the steps, like sample collection, or we call it the specimen collection, and then pressing, drying, stitching, and sewing, and then pasting labels and storage. We'll see the preparation steps one by one. So the first one is the specimen collection. Now, specimen collection is very, very important because it matters like how we are collecting the plant parts, and what part we need to collect, and what is our focus on preparing the herbarium, whether we want to prepare the herbarium of the root, or we want to prepare the herbarium of let's say the rhizome or stem. Even if we are preparing the herbarium of stem or rhizome, we need to still collect the other parts of the plant to make identification of that plant, because these herbariums will be preserved, and somebody may look at those plants maybe after a few years, maybe after 30 years, 50 years, or even 100 years. So it's very important to give the maximum information when it comes to prepare the herbarium. So specimen collection again is very, very important. So we need to go into the field for collection, and we need to collect the plant parts, especially the branches or leaves or flowers or fruits, whatever is available in that season, and make sure that the parts which we collect should not be diseased or should not be infested, and should be clean, and we should always note what you say is so many information at the collection site. So we need to ensure that when we are collecting the uh, specimen, that what is the I mean the altitude of that area. So we need to even carry the GPS meter sometime to check the altitude of that area, and we need to check the whether it was raining that time when we are collecting it, or we need to even look at the physical habitat, like what kind of soil, or the slope elevation, or whether the site is wet or a dry land. And we have to even look at the biological habitat, like what sort of plants are growing nearby to your main plant which you're going to collect. So the nearby plants of growing of nearby or a specimen, and what is the plant community, I should say, and then whether the collection site is an open forest, or is a grassland, or it's just the roadside. So all these things we need to put into the note, you can say the notebook, what we are carrying during our collection. Because when we come back to the lab, we may not remember many things about the plant at the collection site, like how was the stem, how was the leaf, uh, and whether the plant was a shrub or a herb, or it was a tree. So all these information need to be mentioned into the, you can say, the field notebook. Next is the pressing. So once we bring the specimen, or we can say the plant parts from the field, we need to uh, wash it with the water, followed by the 70% of the alcohol. The purpose of treating with the alcohol is to dehydrate, because we need to preserve these samples for a long time. So if there's a moisture in them, then definitely they will be spoiled in the long time. So we need to preserve it. That's the reason that we need to dehydrate them with the 70% of the alcohol. And then we need to neatly put inside the folds of the newspaper, which are water permeable, and then place inside the plastic bags, or we can use the large plastic bag if the sample is large, or if it is a I mean the small plastic bags if so the parts are small. So they are actually pressed in the plant press, or we say herbarium press, and this herbarium press is made up of the wooden frame, or it can have the cardboard ventilators, so that the air is passed through the press. And we also have the blotting paper, and these specimens are actually put in between the newspaper. So we need to cover the sample with the two sheets, or it can be newspaper, it can be blotting paper. And if we have some absorbent, you can say paper, then it will be very good, because they can, you can say, draw the moisture out of it. And then we have to put the thin sheet papers in between the plant material, so that the plant shouldn't stick to that, you can say, the paper in long run. Because if we put it and then we try to uh, open the pressing, let's say after 10 days or 15 days when it is dried, then the plant part should not stick to the paper and should not get damaged. Okay, so we have to be careful in that part. Now next is the drying. So the next stage is the drying of the herbarium, and the herbarium drying can be done in two ways. It can be a natural drying, or it can be done with the oven drying. So if we want to do the drying of the specimen very quickly in 3 to 5 days, we can use the oven, but we have to maintain the temperature between 45 to 50 degrees centigrade, not more than that. Otherwise, it may damage the plant's, you can say, the sample. Even we can use the you can say the natural way of drying, but it will take, of course, a long time. Every day we have to put in the sun every day. It will take around 14 days to dry the sample completely before we go to the next step. Okay, the next step here is stitching and sewing, and this is very, very important, because we have to put the plant, the sample, in a lifelike structure or lifelike arrangement. That means when we put the leaves, we have to turn, when we are putting the branch, having the leaves, we have to turn one leaf like so that we can see the dorsal surface also, ventral surface also. So likewise, we have to put or arrange the collected sample in a lifelike arrangement, and then there should be a space between the leaves. So we should not just when we are arranging, the plant part should not overlay on each other, and there should be enough space on the sheet for a specimen to be put. And later we need a space for label also. So we need to leave some space for label as well when we are putting this plant into the specimen, when we are putting this specimen on the you can say the paper. So the paper which is used for herbarium preparations are actually the special paper we call 100% alpha cellulose herbarium papers, and they are acid free. Why they are acid free? So that they can be retained for many years without any spoilage. And these papers have the pH which is neutral, and then the size of them is like 42 centimeter by 24 centimeter. It's quite a long paper. And then we use glue also to attach these what do you say, this stem or the large part of the leaves or fruits. And but we have to be make sure to use the acid free glue, otherwise it will spoil our paper, the herbarium paper. Okay, large or bulky items may need to be stitched onto the sheet with the sturdy thread or linen. So we need to make sure that the bigger size, or you can say the large part of the bulky, or you can say the bulky part of the branch or the stem, we need to tie, or we need to stitch with the thread. Now, specific parts such as flower, fruits, they can be placed inside the envelope or transparent Ziploc plastic bags, and they can be put, they can be posted on the left-hand side of the herbarium sheet, or they can be pasted in the other places of the herbarium sheet, but definitely not and on the label side. Okay, so for the labeling, we need to have a label which can capture all the information on the plant, like its botanical name, its local name. Let's say in Malaysia, we need to know its Malay name, or Chinese or Tamil name if it is collected from that particular locality. And then we have to have the information on the like date, location, plant features like plant color and plant number when we are collecting it. So we give the number to each plant during the collection, and then photo of the plant should be put behind the herbarium sheet. And labeling, as this is the label. So in the label, as you can see, is a family name, then date, locality, and name of the collector, the person who collect, and then altitude. And even we have to paste the picture of that area behind the herbarium sheet. Then storage, these newly prepared herbarium, they are registered and numbers can be given to these herbariums. And we have the herbarium centers. Even there in other countries, they got some even this government owned herbarium centers, wherein all the information of the flora and fauna is captured in the form of herbarium, and they are systematically actually arranged and preserved. So this storage can be done based on the classification, like it can be based on the family wise. I mean, all the plants of, let's say, Apocynaceae family in one, you can say, place. All the plants of, let's say, Papaveraceae family at one place. All the plants of Leguminosae in that, like Fabaceae, should be on one place. So it can be based on that as well. So storage of the herbarium is done at the dark place, because the light may you can say fade the specimen color. So this is all about the you can say the background of the you can say the preparation of the herbarium, and then now we will see how we prepare it practically. Yeah.
Okay, for this experiment, what we need is the herbarium press. So this is a wooden herbarium press, and we need a 70% of ethanol. Okay, and we need tray, and of course the plant sample to wash it with the 70% of ethanol. And we also need the oven for drying, but in our case we will be doing the natural drying. Okay, so that's all we need to prepare the herbarium.
Okay, in this video you can see the plant part is collected from outside, and you can see this plant part is without any disease part, or it's very proper, and after that it has to be washed in the 70% alcohol to dehydrate, because we need to remove the moisture from the plant so that we can keep it in herbarium form for the long time. So that's the reason that we are actually washing it with 70% of alcohol. Okay, so as you can see that it is dipped into the 70% of the alcohol, which is actually filled in the tray, and each leaf of or leaflet of the plant is actually dipped and washed properly, so that any dirt or any impurity is removed, and the moisture contained also is being removed from this sample. Okay, after washing we need to take it out, and then we need to spread it on the newspaper, and we make sure that the leaves, they don't actually overlay on each other, and we have to spread it in such a way that there is a space in between, and then we have to gently press it so that the sample get dry, and later we transfer it into the newspaper, just to press in between, later for putting into the herbarium press. So here we are putting it in the another newspaper, gently pressing. So that the sample get dry, and this sample is treated with the 70% of alcohol. Okay, so once we dry the sample between the newspaper, after transferring in 2 to 3 newspapers, so that the sample become little bit dry, and then we need to arrange it into the lifelike arrangement. In this case you can see we remove some leaves, because some of the leaves actually during the washing got broken, or some of them were actually found to be bit spoiled, and then we turn some leaves from back side. Okay, so we can see the leaves front and back both, we can say dorsal side also, ventral side also. Okay, that's the reason the leaves are, some of the leaves are turned from the back side. Okay, and then gently press with the newspaper, covered with the newspaper. We need to press it very gently so that the structure don't get destroyed. Okay, and then we need to label it, so that because in this case we are putting so many you can say the plant samples, so we need to later identify the plant samples, so we can put the label here, maybe the plant common name, or even the any other tag for the identification. So after label, we just press it and put some newspapers and the blotting papers or white papers, and then we cover it with the cardboard of the herbarium press. Okay, here is another plant. So for this plant, again, we are actually putting this plant in a lifelike arrangement, and then we are turning actually one of the leaf of this plant so that we can see both the sides. And since there are two samples of the same plant, we can use either one. We have to use only one. We will see later which one comes up better after drying. So not necessarily that both plant sample need to be used, because since it is the same plant, either one can be used depending how they appear after drying. And then we need to label it, and after that we have to put into the herbarium press. Okay, so now we have to keep this plant sample in the herbarium press after labeling, because there are some other plants as well. So we need to make sure that when we take it out, we remember the plant names by the labels. Okay, so this herbarium press need to be, you can say, tightened. That means we need to tie it with the, you can say, belt which is there in this herbarium press. We have to tighten the belt, and we need to leave this for around 2 weeks, because we are doing the natural drying. Alternatively, we can put this into the oven, and then we can get it, the drying process done within 3 to 5 days. But in this case, we are keeping it for 2 weeks. So later, after 2 weeks, the sample will be taken out, and then they will be mounted onto the herbarium sheets, the acid-free herbarium sheets or herbarium paper. So far what we have done, we have collected the sample from outside, and then later we washed it with 70% of the alcohol and for dehydration, and then later we dry it with the help of newspapers, and then we put it into the herbarium press and stack in between the newspapers and the blotting papers and some white papers, and then we tie this herbarium press with a belt, and then we leave it for natural drying. Okay, here you can see the herbarium press, and this herbarium press is open, and then the samples are taken out, the plant samples, and then with the help of glue they are actually pasted on the herbarium sheets. So these are herbarium sheets, acid-free herbarium sheets, and they, with the help of glue, they are acid-free glue, they are normally pasted, and then later they have to be stitched. The sturdy part, you can see the stem part, or even the hard part of the plant should be stitched, so that later during preservation it should not distorted while preserving. So these are the prepared herbarium sheets. Some of the samples here, you can see the shape also, like some are V shape, because Sometimes the herbarium cannot fit into the paper. So these are some of the examples that's it's been prepared before. Yeah. So this is how we prepare the herbarium. Okay, let's see now how to write the report for this experiment. So this experiment, you need to write the, you can say the aim of the experiment, that is preparation of herbarium. Then you need to write the background, like what's the necessity to, prepare the herbarium. And later you need to write the requirement, what is required to prepare the herbarium. Then you need to write the procedure as discussed previously. And then you need to write the observation and uh, discussion and conclusion. So in the observation part, you can write the names of the plants of the herbarium, which you have seen in the video. So we have kept some herbarium in the video, and you can see the individual plant name. Okay, and you can mention few plants. Minimum you can mention three plant name should be okay. And then in the discussion part, you can write about the herbarium, that why we need to use the acid free paper, why we need to use the acid free glue, and what's the importance of you can say the treatment with the 70% of the alcohol prior to putting into the herbarium press. So all these things you can put into the discussion part, and in the conclusion you can write like the herbarium preparations. You observe the herbarium preparation, okay, and then you could uh, uh, list some of the herbarium preparation as shown in the video, the examples that you can put in the conclusion. So I think this will help you to understand this experiment and also to write this experiment. Okay, thank you.